Hi everyone, in today's video we're going to be doing an immunology review, and this is part 1. What is the immune system? It's a part of our body that has a vital role in protecting our body from harmful substances. It can be divided into two, the innate immune system and the adaptive immune system. What cells are involved? So we'll start by looking at the innate system. You have natural killer cells, monocytes, macrophages, and granulocytes. In the adaptive immune system, there's T cells and B cells. The innate system can only recognize a limited number of antigens, but the adaptive can recognize millions. And just so you know, an antigen is any substance that produces an immune response. So looking at the innate system, you can also see it as the first line of defense. And most of the cells that are involved in the innate immune system come from the amyloid cell line. So that begins with the common myeloid progenitor. So the common myeloid progenitor differentiates into many cells, including erythrocytes, also known as red blood cells. But we're mainly concerned with it differentiating into a myeloblast, because from there, that's where you get all your granulocytes, which are your white blood cells involved in the innate immune system. For example, you've got your basophil, you've got your neutrophil, you've got your eosinophil, and finally you've got your monocyte. So the monocyte can migrate out of the blood and differentiate into macrophage and dendritic cells. Most tissues in our body have macrophages. For example, we have alveolar macrophages in our alveoli, we have osteoclasts in the bone, and we have Kupffer cells in the liver. But how do these phagocytes recognize pathogens? And when we say phagocytes, we're talking about these cells of the innate immune system. So phagocytes have these receptors on their surface known as pattern recognition receptors. And in human cells, these pattern recognition receptors are most commonly known as toll-like receptors, or TLRs. And these pattern recognition receptors on the surface of these immune cells have the ability to recognize things known as pathogen-associated molecular patterns, or PAMPs. And examples of these are flagella and a cell wall, both of which are specific to bacteria, specific to pathogens. They're not normal, they shouldn't be found in human cells. So what happens after these pattern recognition receptors, or toll-like receptors, recognize these patterns that you only see in bacteria? So we'll first talk about macrophages and their response. They respond by phagocytosis, which is basically where they engulf and digest pathogens. They also release chemokines, which attract other immune cells to the site of infection. And they also have important links to the adaptive immune system, which we'll discuss in the next video. The next type of cell we'll talk about are neutrophils. They're the most common white blood cell, which is useful to know. Neutrophils are activated by contact with chemokines released from macrophages or dendritic cells, and they release more chemokines themselves, which amplifies inflammation. Let's talk about the classic signs of inflammation. You have rubor, or redness, you have calor, or heat, you have dolor, which is pain, tumor, which represents swelling, and functiola. So these are all Latin terms, and that means loss of function. And these signs all are linked to histamine release as well. So neutrophils, again, they also do phagocytosis. They do degranulation, so the release of their granules secretes antimicrobial substances, and they also generate nets, which stand for neutrophil extracellular traps. The next type of cell we'll talk about are basophils, and they have granules which stain blue. They're quite similar to mast cells, also involved in allergy. And at the site of infection, they release granules which contain several different compounds, including histamine and serotonin, which increase vessel permeability and encourage migration of other white cells. The next cell type are eosinophils, and these granules stain red. The main role of eosinophils is to attack parasites, and they are also associated with allergy. They have granules too, and they contain peroxidases and enzymes which kill cells. Before we go into the last type of innate cell, we're going to talk about something called the MHC, or the major histocompatibility complex. It's a group of genes that code for proteins found on the surfaces of cells that help the immune system recognize foreign substances. And there are two types. Class 1 is found on almost every cell membrane, and these molecules sample peptides generated within the cell and signal the cell's physiological state to effector cells of the immune system, T lymphocytes, and natural killer cells. Class 2 molecules will go into in the next video. So the last innate cell are known as natural killer cells, and it's important to know that they do not come from the myeloid cell line, they come from the lymphoid cell line. What do? They patrol the tissue and the blood, and they read MHC class 1 molecules on cells. And if they detect it as self, then it activates the inhibitory receptor, so it stops them from attacking the cell. However, if they detect something that's non-self, so it's not from the body, this disables the inhibitory receptor, and that causes it to release perforin, which causes a hole in the cell membrane, and that injects granzyme into the target cell and triggers apoptosis, which is cell death. Thank you very much for watching. Next video is adaptive immune system.